Types of cell division. There are two types of cell division, namely mitosis and meiosis. We will start by looking at mitosis. Now, mitosis is a process of cell division in which a cell divides into two daughter cells. This is a parent cell. It divides into two daughter cells, and each of the daughter cells has the same number of chromosomes as the parent cell. You can see here, in the parent cell, we had 46 chromosomes, for example. At the end of mitosis, there will be two daughter cells. And these two daughter cells, each having the same number of chromosomes, that is 46, as the parent cell. So if you take it that the parent cell was diploid, remember diploid means having two sets of chromosomes. At the end of meiosis, the two daughter cells also end up having two sets of chromosomes. So we can see that mitosis ensures that the daughter cells have the same number of genes, because of the same number of chromosomes, as the parent cells. Mitosis usually takes place in the body cells, also known as somatic cells. In animals, the process of mitosis takes place in the bone marrow and Malpighian layer of the skin. Also takes place, that is, in the bone marrow and Malpighian layer of the skin. In flowering plants, mitosis occurs at the tips of the shoot and the roots, where it is responsible for growth. Now, we look at the phases of the process of mitosis. There are 5 distinct phases. There are 5 distinct phases, namely the interphase, prophase, metaphase, anaphase, and telophase. Now, each phase is characterized by specific behavior, appearance, and position of the chromosomes. We look at what happens, the events that take place in each of the 5 phases, starting with interphase. There are 3 main events that take place during interphase of the cell division. These are: 1, there is replication of the DNA. The DNA here is in the form of chromatin. Chromatin is a thin thread-like structure. So during interphase, the genetic material, that is basically the DNA, replicates. So each chromosome then produces an exact copy of itself. So if there was one chromosome, at the end of replication, this single strand will produce a sister chromatid, and the two are joined together at the centromere. That is the first event, the replication of DNA. The second event is formation of new cell organelles, such as mitochondria, ribosomes, Golgi body, and centrioles. The centrioles duplicate. You can see the 2 of them here, and the duplicated centrioles are located just outside the nucleus. So in addition to the formation of new cell organelles, there is a buildup of energy stores in the form of ATP. So within the cell, through respiration, ATP molecules are produced. These help in building up the energy stores which drive the cell division process. Cell division is an energy consuming process. Cell division is an energy consuming process, and as such, ATP molecules are built up in large numbers so that they can provide the energy that is required for this process. Then, after interphase, the process that followed is known as prophase. During prophase, the following processes take place. 1. The duplicated, you can see the duplicated centrioles separate and move to opposite poles of the cell, and the spindle fibers, these are the spindle fibers, begin to form. Spindle fibers begin to form. The other event that takes place is that the chromosomes shorten and become thicker and visible. That is, they become very distinct. And at this stage, each chromosome is seen to consist of a pair of chromatids joined at the centromere. So this chromosome, for example, is made up of 2 sister chromatids that are joined together at the centromere. So this is one chromosome, there's another chromosome here, another chromosome, and a fourth one. Each chromosome is seen to consist of a pair of chromatids joined at the centromere. Another event that takes place at prophase is that the nuclear membrane begins to break down and is completely broken down towards the end of prophase. So those are the distinctive features that occur during prophase. The shortening of the chromatids so that they now become thicker and more visible. The spindle fibers begin to form, and the centrioles having moved to the opposite poles. Then the next event is metaphase. That is, the next phase is metaphase. Metaphase is characterized by the following features. Now, the nuclear membrane has completely broken down and disappeared, and the chromosomes are free within the cytoplasm. The spindle fibers lengthen, and in animal cells, they attach to the centrioles at both ends of the cell. So here we have the centrioles. The spindle fibers have lengthened, and the chromosomes, the third event is that the chromosomes align themselves at the equator of the spindles. So here the chromosomes are aligning themselves at the equator of the spindles, and the chromosomes are attached to the spindle fibers by the centromeres. So it is by the centromeres that these chromosomes are attached to the spindle fibers. After metaphase, the next event is anaphase. Now, during anaphase, the following events take place. The sister chromatids separate at the centromeres. Actually, this is brought about by the shortening of the spindle fibers. So the spindle fibers shorten, and in the process, they split the centromere, thereby pulling the sister chromatids to the opposite poles. That is a distinctive event that takes place at anaphase. The shortening of the spindle fibers, the splitting of the centromere, and thus the separation of the sister chromatids and their migration to the opposite poles. Now, in animal cells, the cell membrane begins to constrict. You can see the constriction here towards the end of the anaphase. You can see this constriction begins at the center. That is, towards the end of anaphase, the cell membrane begins to constrict in animal cells. while at the same time the spindle fibers, after having shortened, begin to disappear towards the end of anaphase. At telophase, the chromatids collect together at the two opposite ends of the spindle fibers. So you can see at telophase, the two sets of chromatids collect together at the opposite ends of the spindle fibers that have now disappeared. The nuclear membrane forms around each. There's a new nuclear membrane that forms around each set of the chromatids. Now, these chromatids are now referred to as chromosomes. They are now fully fledged and functional chromosomes. Another event that takes place during telophase is the division of the cytoplasm into two, eventually leading to the formation of the two daughter cells. Now, as towards the end of telophase, the chromatids will become thin and less distinct. So, after the separation of the chromatids and the formation of nuclear membrane, the cytoplasm divides into two. In animals, it's brought about by the cytoplasmic cleavage, whereby a furrow develops at the center and grows towards the middle, thereby separating the cell into two. Now, in plant cells, constriction is not possible because of the presence of the cell wall. So instead, cell plates form at the center within the cytoplasm, and then these cell plates join to form one continuous cell plate, and later the cell plate grows to separate the two nuclei. Once the cell plate has formed and completely divided the two cells, then new cellular wall materials are deposited, thereby completely separating the two nuclei and the two masses of cytoplasm into two. So in plant cells, there is no constriction of the cell membrane. Instead, there is the formation of a cell plate that grows and extends outward to completely separate the two nuclei into separate daughter cells. Now, mitosis is very important, plays a very important role in living organisms. So these are the roles that are played by the mitosis in living organisms. One, through mitosis, the process of asexual reproduction can take place. That is, it enables asexual reproduction in organisms such as amoeba, bacteria, and some plants. Secondly, it ensures that the genetic constitution of a cell does not change by producing daughter cells that have the same number and type of chromosomes as the parent cell. Thereby, it preserves the genetic constitution of organisms. 3. Mitosis brings about growth in multicellular organisms. By increasing the number of cells in the body, these multicellular organisms become bigger in size. And 4, mitosis brings about repair and replacement of worn-out tissues in multicellular organisms.